Good evening, everyone. Welcome to the session today. Good evening, everybody. I hope all of you can hear me. Yes, so welcome to today's session, everyone. And today, as you know, we are going to talk about gene regulation. So, gene regulation is an important topic from Unit 3, which is fundamental processes, in which you have transcription, replication, transcription, translation, and gene regulation. So, out of that, gene regulation is quite an important topic. Gene regulation basically includes prokaryotic gene regulation as well as eukaryotic gene regulation. Now, in prokaryotic gene regulation, we have basically the operon model. Okay, and in that we have to be very much affluent with a lac operon and tryptophan operon, the working of it and the basic understanding of operon. And then there is eukaryotic gene regulation, how the genes are regulated, how it is controlled. Now, over the years, we have seen many questions coming from this part. Hello, Khushwant. Welcome to the session. Yes, so over the years, we have seen many questions coming from this particular part, and I hope all of you have already prepared it and doing some good practice of the questions. And today we are going to discuss some 10, 11 questions from this particular part. Okay, and we will start with the prokaryotic gene regulation, that is with the operon structures, and over the years, what kind of question has come from there. So we'll start with that, and then we will end with the eukaryotic gene regulation. So everybody, stay tuned and pay focus during the class, and it will be even more enjoyable if you people answer the question. Try to answer it with me, and if you people are actively participating, like for every question I can give you 1 minute or 1.5 minutes for you to answer, and then we can discuss the question. Okay, so let's get started, everyone. Yes, so very first question. Now, this question was actually there in the June exam itself, but it is a relatively very easy question if you have the basic understanding of lac operon, how it works. And also, lac operon is a topic that you have been introduced with from school level. Okay, so although there has been some changes, like when you go in the detail of the topic, some changes, some things changes, some information changes. Okay, but apart from that, the overall process remains same. So, following are few statements made regarding the lac operon. So, A, B, C, D are four statements made. Which of the following option represent the combination of all correct statements? Now, usually questions which are asked from gene regulation are usually this kind of only, where they will be statement based. But when there are statement based questions, then you have to be very much sure whether they are asking you for the correct answers or the incorrect statements. Okay, so this one wants you to identify the correct statements about lac operon. So let's have a look. So statement A says that lac Z, lac Y, and lac A are encoded by a single transcript. Okay, now what do we mean by transcript? Can anyone tell me? What is exactly the meaning of transcript? Single transcript. What do you mean by transcript here? Yes? Anyone? Transcript means the mRNA. Okay, so what is transcript? The thing which is formed after transcription. Okay, so after transcription, what is formed? mRNA, right? So that's why transcript is mRNA. So what it means is that lac Z, lac Y, and lac A are encoded by a single transcript, means encoded by a single mRNA. Okay. Now see, let me draw the structure of lac operon. So suppose this is the lac operon. Okay, so we will start with the promoter, then comes the operator, and then comes the three genes of lac operon: lac Z, lac Y, and lac A. Okay, this is our entire. lack operon. Okay. Now, so basically, what is operon? When multiple genes are put under the control of the same regulatory element, that is what we call as an operon. So in lack operon, there are three genes which are there. Okay, lac Z, lac Y, and lac A. Now, how exactly these genes are expressed? So the RNA polymerase will bind at the promoter. Okay, so binding of RNA polymerase will happen at the promoter. This is our RNA polymerase, and then it will start transcribing the mRNA from this sequence in the DNA. Okay, so RNA polymerase will form a single transcript. So basically, all these three genes in the DNA will be transcribed into a single mRNA. Okay, which will have sections of lac Z, lac Y, and lac A. Okay, so this first statement, which says that lac Z, lac Y, and lac A are encoded by a single transcript, that is correct. Okay. Yes, that you are correct, Maria. So this is our mRNA. Now this mRNA basically contains the coding segment of three genes, right? So three polypeptides can be coded from this single mRNA, and that's why this mRNA is called as polycistronic. Okay. So this is a polycistronic mRNA. So in fact, in all the operons, it will create polycistronic mRNA. Okay. So in prokaryotic cells, we have polycistronic mRNA. Basically, single mRNA can code for multiple polypeptide chains. Okay. Now, what about the translation? So this is what happened during transcription. What about translation? Now, during translation, so second statement says that the three proteins are translated as a single precursor and then processed. Well, this is incorrect because during translation, ribosome will assemble in each of these coding segments, okay, and translate it. Okay, so basically there will be three different ribosomal assembly here, and three different polypeptide chains will be translated. Okay, so from this segment, ribosome will translate polypeptide chain, which is Z. From here, Y will be translated, and from here, A will be translated. Okay, so translation will happen separately. This polycistronic mRNA will have sites for ribosome assembly for all these three polypeptides. Okay, so translation of peptides, the polypeptide chain will happen independently, but mRNA is transcribed as a single transcript. Okay, but translation of proteins will happen independently. Okay, singly at one time. Okay, one cistron of mRNA will code for one polypeptide chain. Okay, like that only, separately only it will happen. So statement B, which says that it will be translated as a single precursor and then processed, that is incorrect. Yes, Sarita and Nagashri, very good. Your answer is correct. So then C statement, in the presence of glucose, lactose can upregulate the operon. Okay, does it ever happen? No. Why it doesn't happen? Because when glucose is present, so what is it? Glucose concentration is high. In that case, CAMP's concentration is low. Okay. Now, CAMP combines with CAP to form the precursor. Okay. So, if CAMP's concentration is low, then CMP-CAP complex cannot be formed. And this CMP-CAP complex, why is it important? Because it is the activator. Okay, so activator is not there. So in the absence of activator, can lac operon be expressed? Yes, it can be expressed, but can it be expressed in a very large expression? No. Okay, so there will be a very less expression because lactose is also present. So lactose will remove the repressor which is bound at the operator region, but because glucose is present, so activator's concentration will be low. In fact, activator will not be formed because CMP's concentration is low, and because of that, in presence of glucose, even lactose is present, still it cannot upregulate the operon. Okay, it will be expression of the operon will be low only. Okay, so statement C incorrect. So as I have told you in the previous sessions also, that's all the question in part. Okay, so So A correct, B and C incorrect. Okay. So A correct, B and C incorrect. So with that, you have already eliminated all the three options. Okay. And statement D, if you see that isopropyl thiobeta D galactoside, that is IPTG, is a gratuitous inducer of lacoperon. That is absolutely correct. Okay. What is a gratuitous inducer? Gratuitous inducer means this is a molecule which is capable of induce the operon, but it is not utilized by the products made by the operon. Okay, so IPTG is one such molecule. So IPTG cannot be broken down by any of these enzymes, these three enzymes, but IPTG can remove the repressor and can induce the operon. Okay, so only statement A and D are correct. Answer is option 4. Yes, very good. Everybody, many people have answered it. Good job. All right, moving on to next question. Yes, thank you for responding. Actually, this is good. Let's keep on doing that. Okay. Next question. E. coli mutants isolated from a genetic screen showed the following classes of mutation. First, point mutation in LAC-I. B. Deletion immediately downstream of the transcription start site of the LAC-ZYA mRNA. C. Duplication of part or whole of LAC-Y. D. Duplication of part or whole of LAC-A. Choose the option which is likely to result in a constitutive expression of the LAC operon. So first of all, we should understand what is a constitutive expression. Okay, so constitutive expression means where there is no regulation on the expression. Okay, see, have you ever seen a fan, or have you ever experienced in a room there's a fan where the regulator does not work? So what happens to that fan? That fan will keep on going at the same speed, right? Because the regulator is not working. So you cannot increase or decrease the speed of the fan. It will keep on going at a constant speed, right? So just like that, if you have a gene which is always expressed in more or less equal amount, then it is called as constitutive expression. All right. Now, what it is saying that there are 4 mutations which are mentioned here, A, B, C, and D. Choose the option which will most likely result into constitutive expression of lac operon. Okay, means constitutive expression basically will mean there is no regulation. Okay, continuous expression of the operon. All right. So when there will be no regulation, when the repressor is not working. Okay. Now let me draw the structure of lac operon once again for you to understand it better. Right. So here we have the operon. Okay. So this is the promoter region. Then you have the operator region. And after that, we have the 3 genes, LAC-Z, LAC-Y, and LAC-A. Okay. Now what happens is at this LAC or O, okay, the operator region, the repressor will bind. Okay. So this will be our repressor bound. Fine. Now, who is coding for repressor? Repressor is coded by another gene which we call as LAC-I. Okay. So LAC-I is present somewhere else in the genome, not within the LAC operon. It will code for the repressor. Okay. And this repressor will be active and it will go and bind with the operator. Okay. Now, for constitutive expression, which needs no regulation, means repressor is not able to bind with the operator. So, by what way repressor cannot bind with the operator? So, repressor, okay. Maria, for you, for this question, your answer is incorrect. Sarita, your answer is correct. Very good. Okay. So, when the repressor cannot bind? Repressor cannot bind with the operator if there is some problem in the repressor molecule. Okay. So if there is some issue in the repressor molecule, then repressor cannot bind, or there is some issue in the operator region because of which it cannot bind. Okay. So there are only 2 possibilities which will not allow the repressor to bind with the operator. Okay. Which is mutation in repressor or mutation in LAC-O, the operator. Okay, do you all agree? So A says that point mutation in LAC-I. Now what is LAC-I? LAC-I is the gene coding for the repressor. So if there is a point mutation in LAC-I, means there is a mutation in R also. Okay, repressor also has the mutation. All right. So if repressor has the mutation, will it be able to bind to O? No. Okay. So mutation in LAC-I means mutation in repressor. Okay. So it will not bind. Fine. So A will result into constitutive expression. Okay. We know A is correct. Okay. So immediately with that, you can cross out option 2 and 3. You are left with 1 and 4. Okay. Now B. Statement B, or the mutation which is saying in B, is saying that deletion immediately downstream of the transcription start site of lag ZYA mRNA. Deletion immediately downstream of the transcription start site. Now, where exactly is the transcription start site? So, RNA polymerase binds here with the promoter, okay, and then from there it will start the transcription. Okay, so immediately downstream. So, first of all, downstream will be this area only, okay, and downstream of the transcription start site. So, if this is +1, so what region is it talking about? It is actually talking about the operator region, area immediately downstream to the transcription start site. Okay, so this means it is talking about LAC-O. So LAC-O has a mutation. So if LAC-O has a mutation, which is the binding site of repressor, will the repressor be able to bind? No. So this also will result into constitutive expression. Okay, so 4 is incorrect. You have got your answer 1. Okay, see, with this you don't even have to check the statement C and D. There is no need of checking the statement C and D. Automatically you got your answer. So anyway, statement C says that duplication or of part or whole of LAC-Y. That means if two LAC-Y regions are present, okay, that will not do anything with the expression of lac operon, because the expression of the lac operon will depend on the regulatory regions. Okay, same with the duplication of part or whole of lac-A, because those are the genes, those are not the regulatory elements. So any regulation, any for constitutive expression or for no expression, there has to be changes in the regulatory segment, not the genes that the operon is controlling. Okay. Right. Moving on to, so our answer is option 1. Okay. Before moving on to the next question, this is an announcement for our new AIML biology, bioinformatics, computational biology batches, which is going to start from 20th Feb onwards. So everybody, you know that bioinformatics is the need of the hour. So if you people are interested, so please contact our toll-free number. You also get the experience of publishing paper, working on hands-on training along with project work. Okay, so all of those things you can get by enrolling with us in our bioinformatic sessions. Okay, so for any further information, please contact our toll-free number. Yes, so Maria, your question, how operator is mutated, then how expression continues? Because operator is mutated means repressor cannot bind with the operator. Okay, so repressor was the one which is controlling the expression, right? When repressor binds with the operator, then there is no expression, because then RNA polymerase will not be able to bind with the promoter region. So if operator is mutated, then repressor cannot bind and RNA polymerase can continuously express the operon. I hope you understood, Maria. See, it's like a magnet you have and an iron piece you have. Okay, so iron will attach with the magnet, right? So if there is some scratches on the surface of the iron, then it will not attach with the magnet. Similarly, if there is some scratch, some plastic or something is present on the surface of the magnet, then it will not attach with the iron, right? So both the ways it will not attach. Okay, similarly, here also the repressor binds with the operator, but if there is any mutation in repressor, then also the binding will be compromised. And if there is some mutation in the operator, then also the binding will be compromised. Okay. Next question. Now, this is a very interesting question which fuses both the operons. Okay, so you have to have a good idea about both the operons, lac operon and tryptophan operon. So, in a strain of E. coli, a fusion between lac and tryptophan operon took place, and the new locus structure is shown below. The strain lacks the wild type tryptophan operon. Given below are some of the potential scenarios. A, B, and C. Choose the option that correctly describes the behavior of the fusion operon. Okay, so the diagram of the fusion operon is given here. Yes. So what do you see in the fusion operon? Okay, so we see that the genes which are the structural genes which are part of the operon are the one which are part of the tryptophan operon, right? Trip E, B, C, D, and A. Okay. But this tryptophan operon genes are under the control of the lac operon, lac I and lac OP. Okay, so basically the regulatory elements are from lac operon. Okay, whereas the structural genes of the operon are from tryptophan operon. Okay, now the question is, given below are some of the potential scenarios. Okay, so how will the working of this particular fusion operon be? Okay, so this fusion operon will work. So it will code the genes of the tryptophan operon. Okay, but the regulation of the operon, like when to turn it on, when to turn it off, that will be similar to the lac operon working, right? Because the regulatory elements here are taken from the lac operon. Okay, so what we have here, statement A says that tryptophan will be synthesized in a medium containing lactose and tryptophan. Okay, so tryptophan will be synthesized in a medium containing lactose and tryptophan. So here actually tryptophan will have no effect. Okay, because the regulatory element are from lac operon. So the regulation of that will be completely under lac operon. Okay, so when there is lactose, so lactose will be converted into allolactose, and allolactose will bind with the repressor and remove the repressor. Okay, so removal of the repressor from lac operator region will result in the synthesis of the operon genes. Okay, so tryptophan will be, and these genes will do what? They will ultimately participate in the conversion of chorismate to tryptophan, so in the tryptophan synthesis. Okay, so therefore when lactose is present, tryptophan will be synthesized in the medium. Okay, but tryptophan being present and or absent has nothing to do with the working. Okay, so statement A correct. B, tryptophan synthesis will be repressed in the medium containing glucose. Okay, now see, I told you the regulation will be just like lac operon. So in lac operon, what happens when glucose is present? CAMP's concentration is low. CAMP complex, which is the activator, is not formed, right? So CAP complex will not be there. Okay, now when it is not there means activator is not there. Okay, and in absence of activator, the expression of the operon becomes very low. Okay, so tryptophan synthesis will be repressed in a medium containing glucose. Absolutely correct. Okay, so this will happen because here the genes are required for tryptophan synthesis. So if the operon, if this particular operon's expression is repressed, means tryptophan synthesis will also be repressed. Now, so A and B both are correct, right? So with that, you already got your answer even without checking statement C. But we'll check it here. So tryptophan synthesis will take place only in the absence of sufficient tryptophan in the medium. Now, this statement is the actual control on tryptophan operon. So where tryptophan itself acts as the activator. So in case of the regular tryptophan operon, the repressor is not able to bind to the operator. Repressor will only bind with the operator when the repressor is bound with the tryptophan molecule. Okay, so binding of TRIP-R with tryptophan makes it an active repressor, and then this active repressor can go and bind with the operator to repress the tryptophan operon. Okay, This is the regular control. But here, are we having any trip repressor here? No. Okay. So tryptophan synthesis will take place only in absence of sufficient tryptophan in the medium, will not happen because the control will be by lac operon. So it will behave with the presence or absence of lactose and glucose, not with the tryptophan. Okay. So that's why C will not happen in this particular fusion operon. Answer will be A and B. I hope it's clear to all of you. Moving on. Next question. This is a very interesting question, and this is actually a quite easy question if you have a good understanding of translation process. So eukaryotic mRNA shown schematically in the diagram below has 5 ribosomes carrying out translation. Which one of the following statements about this polyribosome complex is true? Okay, so the structure that is shown here is actually a polyribosome, okay, or which is also called as a polysome. What happens in polysome? That a single mRNA can be translated simultaneously by multiple ribosome complexes. So at a time, a single mRNA can be translated into different protein molecules. Okay, so that can this answers the immediate need of any protein by the cell. Okay, so if the mRNA is there, so immediately it so the protein coded by it, the demand for that protein coded by it can be met very fast here. Okay. Now ribosomes, when it translate the mRNA, it will always move in which direction in the mRNA? 5 prime to 3 prime direction. Okay. That means what? That this ribosome 5 is the one. So movement of ribosome is happening from here to here, from 5 prime to 3 prime. Okay. So definitely that means what? That this ribosome, so near this 5 prime end only, there will be the initiation codon, or I will write it as the start codon, whereas near the 3 prime end only will be the stop codon. Okay. Now ribosome, it's not that ribosome, so when we talk about this ribosomal assembly, okay, so it does not happen that ribosome will come and join anywhere. Ribosome assembly will always happen at the start site or the initiation codon. Okay, so if this is the initiation codon, so here only the ribosome will assemble. Okay, and then this ribosome will move from 5 prime to 3 prime direction, translating the mRNA. Okay, so suppose this ribosome has come here and it has translated this region. Okay, so this much polypeptide is ready. That means there is nothing here. Okay, since there is nothing here, that is the initiation codon is free, so a second ribosome can come and join. Okay, joining of this second ribosome will do what? Then again it will start translating. Okay, so this ribosome will now move to this place, this ribosome will come here. Okay, so here the ribosome came, the first ribosome came here. Okay, and the second ribosome came here. All right, again the initiation site is free, so a third ribosome can now come and assemble here. Okay, so this is how the ribosome will keep on moving. So basically the ribosome which has moved most to the 3 prime end will have the longest polypeptide chain, whereas the ribosome which is most close to the 5 prime end of the mRNA will have the smallest polypeptide chain. Okay, do you all agree? Yes, now let's see the statements here. The mRNA was being transcribed when the first ribosome started translation. So what it is saying, that when the first ribosome started translation, mRNA transcription was still continuing. Is it possible here? Answer is no. Why not? Because in the question it is clearly mentioned that it is an eukaryotic mRNA. For eukaryotic mRNA, this is not possible because eukaryotic mRNA is transcribed in the nucleus and translated in the cytoplasm. Yes, this statement would have been correct if we were talking about prokaryotic mRNA, but this is eukaryotic mRNA, so statement 1 is incorrect. 2 says that ribosome 5 is nearest to the initiation codon. As I told you, initiation codon will be nearest to the 5 prime site. So if ribosome 5 is on the 3' side, so it is not nearest to the initiation codon, it is in fact nearest to the stop codon. Okay, so this statement is also incorrect. Fourth states that polypeptide attached to ribosome 4 is longer than the one attached to ribosome 3. Okay, so as I told you that ribosome will keep on moving from 5' to 3' direction and translating the mRNA. So definitely the ribosome, which is the fifth ribosome, will have the longest polypeptide chain among all. Next will be the ribosome 4. Next will be ribosome 3, then 2, and finally 1, the smallest chain. Okay. So definitely the ribosome number 4 will have a polypeptide chain longer attached to it than ribosome 3. So this is the correct statement. Statement 4 says that all the ribosomes have incorporated the carboxyl terminal amino acid. Now, when we talk about translation, so the amino acid which are added, okay, how they are added? So amino acid synthesis. So if I write it as the polypeptide chain, okay, so the polypeptide chain means there will be multiple amino acid residues which are joined by a peptide bond, right? That is what is a polypeptide chain. So in this polypeptide chain, this synthesis of polypeptide chain starts from the amino terminal to the carboxyl terminal. Okay, so the first amino acid added to the polypeptide chain will always have amino terminal free, whereas the last amino acid added to the chain will have carboxyl terminal free. Okay, that is why we always write down polypeptides also from N to C. Okay, so the statement that all ribosomes have incorporated the carboxyl terminal amino acid, all of these ribosomes, is incorrect, because carboxyl terminal amino acid will be added at the end, just before the second last codon. So last codon will be the termination codon, so second last codon will be the amino acid at the carboxyl terminal. So all of these ribosomes have incorporated the amino terminal amino acid, but not the carboxyl terminal. Okay, so this statement is also incorrect. Answer is option 3. Okay, so this is a very fundamental question. If you have the idea how RNA translate, how the translation moves, okay, during translation, how the ribosome slides, or in which direction the ribosome moves in the mRNA, or how the polypeptide chain is synthesized. Okay, so it will be very easy for you to answer. Okay, moving on to next question. A new strain of bacteria was created by introducing an artificial operon to allow bacterial cell to grow in the presence of iron. The Fe++ operon consisted of genes that are made up of cells capable of tolerating increased iron. Okay, so what is it? Fe++ operon consisted of genes that make the cell capable of tolerating increased iron. For efficient functioning of this operon, the following desirable features were considered. Which one of the following can be used to develop this particular operon? Okay, so let's first understand what is the functioning of this operon. So this operon is saying that it will control the genes. So let's say that there are XYZ, 3 genes. Okay, so operon means there will be a operator. Before that, there will be a promoter. Okay, and then there will be the genes. Okay, let's say there are 1, 2, and 3 genes. Okay, and what is the function of these genes? So these genes will make the cell capable of tolerating increased iron. Okay, so basically if you have a operon like that, okay, so when will you want the operon to express itself? So you want the operon to be expressed when iron concentration is high, right? So means we want the operon to be active. So operon has to be active when iron concentration is high. Okay, because when iron concentration is high, then only the cell will need help to tolerate those high iron concentration. So in that time, when the operon is active, so it can create, it can transcribe and translate these genes, and that gene, those gene products will help the cell tolerate this increased iron concentration. Okay, now when iron concentration is already low, do you want the operon to be active? No, because that will be such a waste of cells resources. Okay, because iron concentration is not high, why do cell need resources, waste its resources to transcribe and translate these genes 1 2 3? Okay, so the operon has to be inactive when iron concentration is low. Okay, so iron concentration high, we want the operon to be active. High iron concentration low, we want the operon to be inactive. Okay, so this is the functioning that we are looking for. Now let's see this option. So statement 1 says that the repressor protein which is active when bound to iron. Okay, so a repressor protein active. Active means then the repressor is able to do its job, means it is able to repress the operon. Okay, so when iron concentration is high, then only iron will go and bind with the repressor. So binding of iron with the repressor, if it is making it active, means then it is allowing the repressor. So repressor plus iron, okay, it is converting it into active. And then this repressor plus iron can go and bind with the operator. Okay, so if it is binding with the operator, will there be transcription? No. Okay, so this is not possible. This is not the control that we are looking for. Okay, next, an activator protein which is active when not bound to iron. Okay, now what we need, so the second statement says that an activator protein. So activator protein will activate the operon. Okay, will increase the expression, which is active when not bound to iron. Okay, so not bound to iron, it is active, means activator alone is able to bind here and it can transcribe. Okay, so when activator is bound with iron, then activator cannot bind. Okay, that again is not the situation we want. Otherwise, in this particular case, what will happen, that even when even when iron is not present, then also this operon is on, but when iron is present, the operon will be turned off. Okay, the exact opposite of the function that we want. Again, not possible. Third says that the repressor protein which is inactive when bound with iron. Now this is making sense. So repressor protein which is inactive when bound with iron. So basically there will be a repressor protein which is active and binding with operator. But when it binds with iron, okay, then it becomes inactive. Okay, and so that repressor plus iron is, it dissociates from the operator. Okay, so as it dissociates from operator, then it can turn on the expression of these genes. Okay, so when iron concentration is high, then iron will convert the repressor into an active form, remove the repressor from binding with the operator, and turn on the operon. Okay, so iron concentration high, in that case operon will be active because repressor will be removed. When iron concentration is low, okay, then repressor again will not bind with iron when iron concentration is low, and in that case repressor will go and bind with the operator, and that will turn off the operon. Okay, so option 3 seems correct. Let's see 4 also. A repressor protein which is active constitutively. So if we have a repressor protein which is always active, so in that case, the situation will be that the repressor will always bind with the operator and this operon will always be turned off. Okay, but we don't want it because we want the genes to be expressed when the iron concentration increases. But if this repressor is constitutively active and we cannot make it inactive, so in that case, this regulation on the operon will not be there. Okay, so answer will be option 3. Option 3 is our answer. Right. So moving on to next question. I hope all of you are following. Can I have some responses? Sarita, are you there? Not getting any answer from you. Sarita, Maria, Nagashri. Keep responding. Yes. So are you all following? Okay. Let's have a look at this question. In context of lacoperon, if two bacterial strains P1 and P2 with the genotype OCI plus Z minus and O plus I minus Z plus respectively were used to produce Mero-diploid daughter strain daughter strain D. Which one of the following statements correctly predict the expression of Z gene, that is beta galactosidase activity, in all the three strains? O plus, I plus, and Z plus denote the wild type allele of the respective genes. Okay, so if O plus, I plus, and Z plus are the wild type allele, means OC, I minus, and Z minus are the mutant forms. Correct? Okay. So let's write down the genotype of each of these strains and see what will be its expression. Okay. So I'll start with P1. So P1's genotype is given as OC, I plus, and Z minus. Okay. So I plus, I will code for R, LAC-R, the repressor. Okay. Now this repressor will go and bind with the operator. Will the repressor be able to go and bind with the operator? No, because O is mutated. Okay. Fine. But before that also, what do you see? Z is minus. So it is a mutant allele of beta galactosidase gene is present. Okay. So if Z itself is mutant, so beta galactosidase will not be produced. We don't even have to look at the regulation, but beta galactosidase will not be produced because the gene itself is mutant. Okay. So P1 will for sure be no expression. This is very easy. P1 has no expression. Okay. Now with P1 no expression, you can only eliminate option 4. All other three has P1 no expression, no expression, no expression. Now let's have a look at P2. Sorry. Right. So what about P2? Now P2's genotype given is O plus I minus Z plus. Okay. Now here Z is plus, means wild type allele is present, so it can code for beta galactosidase. Now what kind of expression will be? So for that we have to look at the regulatory region. So LAC-I is minus, means will it code for the repressor? No. Okay. So if there is no repressor, means nothing to bind with the operator. Okay. So in that case, what kind of expression it will have? It will have a constitutive expression. Regulation of the operon is there by repressor. So if the repressor itself is not there, okay, so the type of expression will be constitutive. So P2 will have constitutive expression. Now with that information, again, you can eliminate option 3. So P2 constitutive in there, option 1 and 2. Okay. Now you have to look for the D strain, which is formed by combining P1 and P2. So let's write it down here. Okay, so I'm just clearing out P1 so that we have space to write the D strain. Okay, now D strain is a Mero-diploid strain. Now what is Mero-diploid? Mero-diploid means it is partially diploid. Okay, how this partially diploid strain is produced? By combining both P1 and P2. Okay, so basically this D strain will have the genotype of, it will have the genotype of P1 in its bacterial DNA, while it will have the genotype of P2 in its plasmid DNA. Okay, or it can be vice versa. Okay, basically it will be partially diploid, so there will be 2 copies of these genes present. Okay, but all other genes will be present in a single copy. Okay, now in this particular case, the situation will be, so first of all, this DNA which has O mutant, okay, Z is also mutant. So this particular DNA will not even code for any beta galactosidase. Which DNA will code for beta galactosidase? This one, because it has Z plus. Okay. And here O plus is there, the wild type of the operator region. Okay, but the DNA, the lac I which is present here is minus. Okay, but what do we know? That in lac I doesn't have to be present in the same DNA for the regulation. Why? Because LAC-I directly we don't need. We need the repressor which is coded by LAC-I. Okay. So in this particular D strain, what will happen? That this LAC-I will produce the repressor. Okay. The repressor being the protein molecule, it will diffuse through the cell and go and bind with this operator. Okay. This repressor cannot bind here because O is mutated. Okay. This repressor will go and bind with the operator here. Okay. Binding of repressor with the operator. Okay. So that will stop the expression of Z. Okay. But what will happen in presence of lactose? Then lactose will be converted into allolactose, repressor will be removed, and Z will be expressed. Okay. So what kind of expression will it be? This is the normal situation, how the expression works. Okay. Which is an inducible expression. So strain D will have an inducible gene expression. Okay. So with that, you can eliminate option 2 and 1 is your answer. I hope everybody understood this. Yes, absolutely correct, Sarita and Maria. LAC-I can work in trans. It can be present anywhere else in the genome. It can be present in a different DNA molecule also, just that it should be working. Okay. Next question. Now, this is also a statement-based question. And to answer this statement-based question, you just have to read the question properly. So the LAC-I and TRPR repressors bind with their ligand, allolactose and tryptophan respectively, resulting in alteration of their DNA binding properties. The following statements were made about the mechanism of LAC-I and TRPR binding with DNA operator and the regulation of gene expression in E. coli. So in this matter, there are 4 statements given, A, B, C, D. Which one of the following option represent a combination of all correct statement? Okay. Now, before answering it, I'll just give you a brief recap of what are lac-operon, tryptophan operon. So we have been discussing about lac-operon till now. So lac-operon, you know, is an inducible operon and the regulation is with the help of repressor. So repressor will go and bind with the operator, and when lactose is present, then lactose will bind with will be converted into allolactose. Allolactose will bind with the repressor and it will be removed. Okay. So then the lac-operon is induced. For tryptophan, it is a repressible operon. So the working is different. So when tryptophan concentration is low, okay, then the repressor here, TRPR, cannot bind with the operator. Okay. So tryptophan, the genes of the tryptophan operon will be synthesized and tryptophan synthesis will continue. When tryptophan concentration increases, okay, or becomes too much, then we have to turn off the operon. So how it will happen? So the tryptophan itself will act as a molecular signal. It will bind with the repressor, make it active. Now the repressor plus TRPR will be able to go and bind with the operator, and that will turn off the operon. Okay. So basically binding of lactose with LAC-R or binding of allolactose with with the LAC-I product will induce lac-operon, whereas binding of tryptophan with TRPR will repress tryptophan operon. Okay. So you can see here statement 1 that allolactose binding with LAC-I leads to poor binding of the LAC operator, whereas tryptophan binding to TRPR leads to its better binding to the TRP operator. So statement A, absolutely correct. Now, what about statement B? Allolactose binding to LAC-I leads to induction of the lac-operon. Yes, because allolactose binding to LAC-I will make it inactive, so that will induce the lac-operon. Whereas tryptophan binding to TRPR will lead to repression. Yes, because tryptophan binding to TRPR will make it inactive. Because I will write it here. So allo lactose plus LAC-I. Okay, what it results into? inactive repressor, whereas tryptophan plus TRPR results into active repressor. So this statement is also correct. So A and B both you have understood is correct. Okay, now again with that, if you scan the options given, so there is B and C, you can automatically eliminate. So you have A and B only, or A, B, D. Okay, so there is actually no need of analyzing the statement C, but we will do that also. But later, let's check D. Okay, so D says that binding of allolactose and tryptophan to lac-I and TRPR respectively leads to activation of their corresponding operon. Binding of tryptophan with TRPR will result into activation of corresponding operon. Will it result into activation of operon? No. First part, that is allolactose binding with lac-I, will result into its activation of the lac operon. That is correct. But binding of tryptophan with TRPR will result into inactivation of its operon. Okay. So, however, in operon regulation, availability of tryptophan also results into attenuation mediated transcriptional termination, leading to an overall effect of repression of the tryptophan operon. Okay. So, the first part of the statement itself is incorrect. Okay. However, the second part, that tryptophan is there to control and do the transcriptional attenuation also, that is right. That is a secondary regulation which is there, but that is only for fine tuning the regulation. It cannot cause the overall regulation. Okay, only it can fine tune the regulation. All right, so D is incorrect. I hope you understood, Nagashri. But first few codons will form where? In what case first few codons will form? If repressor is there, yeah. Okay, so repressor is binding, yes, but what about the polymerase which has now moved on to the leader peptide region? Okay, so for that we have the attenuation mechanism where transcription starts normally, but it is abruptly halted before the first gene of the operon is transcribed. Okay, so for that we have in tryptophan operon we have the attenuation mechanism. So attenuation mechanism is not for the course control. For course control we have this repressor. Okay, attenuation mechanism is there for only the fine tuning the regulation. So first few codons will form, but because of attenuation, the transcription will stop in between. Okay, so the RNA polymerase will leave. So it will have a incompletely formed small RNA, small mRNA, and that incomplete mRNA and short mRNA will be degraded. Am I clear? So answer will be option A. Only statement A and B are correct. Okay. Now moving on to the eukaryotic gene regulation. Okay. Now see this question. Okay, I have added here because last 2-3 sessions, some or the other question related to heat shock and how the gene expression changes during heat shock. Okay, that has come. So during heat shock, mammalian cells shut down global protein synthesis while inducing heat shock proteins. Okay, which is HSP. The possible molecular regulation that could explain the phenomena are, okay, so what happens is during the heat shock, so it's a emergency for the cell, so cell will try to code only for those gene products that will allow the cell to survive the heat shock. Okay, because survival is the priority. All right, so all other mRNA synthesis will be blocked. Okay, how it is blocked? So it is not possible for the cell to selectively degrade all the mRNAs. Okay, instead what the cell do is cell will degrade the translation factor which is required for bringing the mRNA to the ribosome. Okay, So what is that particular protein factor that will be degraded? That is the cap binding protein, which is EIF4E. So EIF4E is part of translation factor, translation initiation factor EIF4F. Okay, and this EIF4E will do what? It will go and bind with the 5' cap of the mRNA, and it will help in bringing the mRNA to the ribosome complex. Okay, so cap dependent translation of most mRNA is affected during heat shock due to denaturation of this cap binding protein. Okay, so EIF4E will bind with the cap of the mRNA, 5' cap of the mRNA, then the mRNA will be taken. So that is the general process which is followed during translation by all eukaryotic mRNAs. Okay, so when we have to stop, when the cell has to stop the translation of this regular mRNAs, so it degrades EIF4E. Okay, so that there is nothing to bring the mRNA to the ribosome. Okay, now how the HSP mRNAs will be translated then? So translation initiation of HSP mRNA take place through their internal ribosome entry sites. So this HSP mRNAs will have an internal ribosome entry site, which is called as IRES, and ribosome will attach with those HSP mRNAs through this internal site. Okay, it doesn't have to go through the 5' binding cap. Okay, so it can internally go there, and only this HSP mRNAs will be translated, that so that the cell can survive the emergency. Now once the emergency is gone, then again EIF4E will be made in the cell, and the regular translation will start. Okay, so which of the following set is correct? So statement B and C are correct. Okay, next question. Now this is a type of question which has come again multiple times, which basically is a deletion mapping question that helps us in identifying the activator binding site or the repressor binding site. Okay, so in order to identify the regulatory region of a noble promoter sequence shown above, four 150 base pair deletion constructs were made in a luciferase reporter system as indicated above in boxes A to D. After transfection, the observed level of promoter activity percentage are analyzed by luciferase assay of all the constructs is indicated in the right of the figure. Identify the best correct combination of regions in the options given below that indicate the presence of a positive and a negative regulatory element respectively. Okay, now see, these are the deleted segment. Okay, so what do you have to understand first is that if you have a positive regulatory element, okay, so positive regulatory element will do what? It will increase the expression. Okay, so if this positive regulatory element, if deleted, what will happen? Expression will decrease. Okay, similarly, what about negative element? So if you have a negative regulatory element, okay, what it will do? It will decrease the expression. Okay, so if deleted, if this negative regulatory element is deleted, what it will result into? Increase in the expression. Correct? Okay, so what do you see here? That A, when this segment is deleted, promoter activity decreases. Okay, so if expression decreases, so what it can be? It must be the positive regulatory element attachment site, so that when it is deleted, promoter activity decreases. Okay, similarly in here, okay, 140%, the promoter activity increases to 140%. Okay, so what should be this C? This C should be the negative regulatory element, because when negative regulatory element was present, then promoter activity was 100%. When it is deleted, promoter activity goes beyond 100%. It became 140. Okay, so N, the negative regulatory element, must be binding at C. Okay, so what are the positive and negative regulatory element? So A and C are the binding sites of positive and negative regulatory element respectively. So answer is option B. Yes, so I have one more question here, and this is from eukaryotic gene expression. And again, in last 4 or 5 years, 2 or 3 questions, similar question has come. Okay, now as you know, at the end of the session, you will get 5 more questions with solutions from the same topic. So there, I have included one more question, which is very much similar to this only about the eukaryotic gene expression. Okay. So following statements are given, you have to identify the correct statements. Okay. So first says that usually regulated at the level of initiation of transcription by altering the chromatin architecture. Now this is absolutely correct for eukaryotic gene expression. So in eukaryotic gene expression, since the genes are present in the chromosome and chromosomes are already in a very complex chromatin structure, so the regulation, the first level of regulation of the gene expression is at the transcription level, and that happens by uh, whether the gene is accessible to the transcription machinery or not. Okay, so that's why euchromatin region, which is the less compact region, usually have the genes which are always expressed, whereas the heterochromatin region will have the genes which are never expressed. Okay, so heterochromatin means very compactly coiled. Okay, so it is regulated at the level of initiation of transcription by altering the chromatin architecture. That is what happens. So if a gene has to be expressed, so that particular region, the chromatin architecture of that region will be relaxed actually. Okay, so that the DNA will become loosen up and the DNA will be accessible by the transcription machinery and the gene can be expressed. Statement A correct. B says that eukaryotic genome is divided into domains of insulator elements. Absolutely correct. We know that a gene is divided into introns and exons. Okay. So introns are the insulator elements. Okay. So B is correct. C, chromatin remodeling complex binds with the promoter of the gene in sequence specific manner. Okay. So chromatin remodeling complex binds to relax the chromatin architecture. True. But it does not bind with the promoter. No, promoter is the binding site of RNA polymerase here also. Chromatin remodeling complex will bind where? It will bind with the enhancer sequence, which is the binding site for them. Okay, not the promoter. All right. So that's why statement C incorrect. D, architectural protein regulate gene expression by promoting DNA bending. Absolutely right. So basically, if this is a gene, okay, so we have a gene, and if this is the promoter of the gene, okay, so many nucleotide away, there will be the enhancer sequence. Okay, so enhancer sequence is the site, as I told you, the architecture, the chromatin remodeling complexes or the transcription factors will bind. Okay, so from this far, how it will regulate? The architecture modifiers will bind here. Okay, and that will bend the DNA. Okay, bending the DNA will bring the enhancer close. Okay, and then it can work. So this is correct. E, the chromatin remodeling complex can alter nucleosomal structure but cannot displace them. Now this is incorrect, okay, because chromatin remodeling complex not only can alter nucleosomal architecture, it can completely displace them. Okay, it can completely remove the nucleosome so that DNA becomes very easily accessible. All right, and when the DNA's function is done, okay, when the gene expression is done and no longer expression is needed, then again the nucleosomal assembly will be reassembled. Okay, so statement E is incorrect. So what are correct? A, B, and D. These are the statements which are correct regarding eukaryotic gene expression. Yes, the architecture modifiers, they are the HMG proteins. That's correct, Sarita. Okay. So very good, everyone. And you made the session active also. So this is a similar question to the one that we have already seen, and you can try to solve it on your own. Okay, this will be like a homework for you. Along with that, our team will be adding the questions. Okay, so in the link, and you can download the link, and all the 5 questions will be there along with their explanation. Okay, so please go ahead and do it, and all the best for your exam, everybody. And I hope all of you are doing great, and great preparation is going on. And don't forget, if you want to do some course, some certification in bioinformatics, Biotecnika is there with you. Along with the training, we also will help you in publishing papers and doing project work. So if for any further information, please give us a call in our toll free number. Thank you everyone for today's session. Have a great evening. Bye bye.